Hello, I'm Regine Mortazevi, and I'll be presenting Denaturant versus Nurture, the unfolding of myoglobin. For the introduction, to begin, we must explain what myoglobin is. It is an oxygen-binding protein in muscles. Its importance lies in the fact that oxygen lacks solubility and can't reversibly bond to proteins readily to transport muscle cells. Myoglobin contains a heme, shown in the bottom left, which contains a coordinated iron ion, which has a high affinity for oxygen. To properly bind the protein needs to be in its native or folded state. The denaturant, guanidine hydrochloride, unfolds the protein, which prevents the binding and release of oxygen, which is why we are studying it today, to observe its reaction and impact on energy. When unfolded, the protein can no longer bond with oxygen, and if already binded when unfolded, the process is no longer reversible, which, oxygen not in muscles, not the greatest thing. We'll use a UV visible spectrophotometer to collect the absorbance data of the native versus unfolded protein and use those values for future calculations that we'll get to if you guys stay tuned. The spectrophotometer we used for this experiment is a Vernier SpectroVis Plus. It is a single beam light source instrument, meaning that one ray is projected through the sample for photons to be absorbed. The detector of this instrument is a charged coupled device. This type of device is a sensitive photon detector. When photons reach the areas on the detector called pixels, they're converted to electrons which are stacked. This stacking leads to an ability to measure intensity. The data then collected through the detector is converted into absorbance for the spectra used in this experiment. Now to get into the solution preparation as well as data collection. Do not fear the giant table. It is simply the guidelines followed to create the samples at increasing concentrations of denaturant. We used precision measurement tools to create each sample, ensuring accurate molarity between samples for the calculations that will follow. Once made, the samples were allowed to sit for about 20 minutes. That way, the guanidine had sufficient time to unfold the myoglobin. Prior to inserting the samples into the spectrophotometer and collecting data, we used a blank, or just buffer, to collect a background. The collected data then will be shown in the next slide. In the spectrum, we have limited the samples shown to 5 that display the trend. The highest absorption peak corresponds to the solution containing no denaturant, with gradual decreases until a sudden dip in absorption at the higher concentrations. We isolated the absorption values at 400.9 nanometers to use for calculations and other conclusions. The figure shown is the absorption at the peak wavelengths of all 19 samples. This explains the overlapping of peak signals. The graph is essentially a titration curve. The plateaued portions are below the baseline concentration of denaturant, which would have essentially no unfolded protein, and the point past the maximum concentration of denaturant needed to unfold most, if not all, the proteins. This is where we will choose our absorption of native and unfolded protein values. In the very center is where there is an equal amount of native and unfolded proteins. This value is called C subscript M, which we'll be referencing later. For this, we will be using the linear position data points. And those are the concentrations of guanidine hydrochloride 0.9, 0.95, 1, 1.05, 1.1, 1.15, 1.2, and 1.25 molarity. Now, a brief summary of the relevant equations and their variables before we get into the final results. As mentioned before, we selected values of absorption in fully native or unfolded solutions. Those are used to find the equilibrium constant of the denaturing reaction with respect to the peak absorption of each sample. That's the AN and AU in the first equation. The equilibrium constant is then used to solve for the free energy of unfolding, the first part of the second line of equations. The graph free energy then allows us to use Excel's linest function to determine the slope, rate of reaction, and the free energy of unfolding in the absence of denaturant. That's the third equation. The following two equations are the fraction of unfolded proteins in solution experimentally and theoretically, which the theoretical was found using Excel's solver function to minimize the chi-squared value of the two variables. The equation for the theoretical fraction was determined by solving for the equilibrium constant, the second equation in the second line, and plugging that into the experimental fraction equation. We found that the free energy of unfolding decreased at higher concentrations, which is indicative of the loss of noncovalent bonds that held the folded protein together. You can see the graph for that on the right. The experimental and theoretical fraction of unfolded proteins were quite similar, meaning that our results were close to ideal conditions. The experimental and optimized slope and concentration at which equal native and unfolded proteins were present are shown in the table on the bottom right for a more quantitative comparison, as well as the fraction graph of comparing the theoretical and experimental. Now for the discussion. The SORT band refers to the high absorption that occurs around 400 nanometers, which corresponds to the blue region of the visible spectrum. We observed that with an increase in denaturant, there was an increase in absorption and increase in energy due to a higher conjugated heme when unfolded. This is shown in the figure. When we say increase in energy because of the conjugated heme, we mean that the unfolded structure is able to absorb at higher wavelengths, which explains the tilt to the left of the peaks as the concentration increases, top to bottom. The native stability of the protein can be analyzed using entropy and enthalpy. The unfolding of myoglobin leads to a higher entropy state, which is desired, but also requires increased enthalpy to break noncovalent bonds, which is undesirable. The higher entropy is because we are removing order and stabilized structural qualities of the protein. So while entropy pushes towards disaster and unfolding, the energy required to break the noncovalent bonds that give this structure and stability keep it at bay. Our experiment essentially increased the push of entropy and decreased the energy required to unfold, as is seen in our free energy calculations. The rate of denaturation, m, displays a titration-like pace, needing to meet a certain barrier and then leading to a rapid reaction. This is the slope found from the linear section of the graphed free energy of unfolding against the concentration of the guanidinium hydrochloride. To conclude, this experiment made conclusions on the energy and reaction characteristics of the unfolding of myoglobin. We know that energy decreases with further unfolding, and conversely, the absorption energy increases. With these results, we can ask the audience, is unfolding nature or nurture? Thank you for watching, and I hope this was informative.